Recording started.
Okay, so welcome to everyone. We just started the recording. We hope that this presentation will be posted on some website in the next week or two, and we'll distribute that information for anyone who wants to hear the information and could not make it. So I just started to say that we discovered this mutation about a year ago along with a few other groups. And we found that this mutation was mutated in virtually all of the tumors that we were calling at the time small cell carcinoma of the ovary hypercalcemic type. Let's see, where are we here? There we go. The unique thing is that these mutations do occur in other tumors, as shown on this figure, but they don't occur as such high frequency, and they also have a different type of mutation. So these mutations are called non-missense mutations, which means there's a large change in the DNA sequence such that pieces of DNA are either removed or inserted. Those are called insertions and deletions. Or the gene gets truncated and it becomes too short and the protein becomes too short. Those are what we call non-missense mutations or frame shift mutations and nonsense mutations. Most of the mutations that occur in other tumors are called missense mutations, which means that the gene and protein are usually about the same length, but one amino acid has been changed, and so the sequence is different, and sometimes that will result in an important change in function, and sometimes it won't, and that's all the blue bars that you see. And so this is a very unique disease in that all the mutations are really what we call functional mutations, shown in orange here. And they're very uncommon in other tumor types. You can see only 3% of almost 5,000 tumors have this gene mutated, whereas in this tumor type, it's nearly 100%. On the next slide, we show, I'll just show you here, these are some gene expression studies where we show that the gene is truncated because of the mutation. In the middle panel here, you can see where there's no band. I think you could probably see my mouse on the screen. You can see there's a loss of protein in the small cell tumors. These are regular ovarian cancers called high-grade serous carcinomas, and they have retained the protein, and these are our positive and negative controls. And so the mutation results in a loss of protein expression. This is called a Western blot. And here's a tissue section. These are actually tumor cells. In the middle, you can see a positive control. This is a small cluster of tumor cells that's very brown because it is expressing the protein. This not-so-brown area here is actually tumor cells that do not express the protein, and the brown part here in the middle is a positive control. These are non-tumor cells that do express the protein. So we've shown that the mutations cause loss of protein expression, which leads to loss of protein function. Here we also show in our original paper that if we knock down the gene on the top line here in just some regular sort of kidney cells, the tumors grow quicker. And if we put the gene back in this lung cancer cell line that doesn't have the gene, the tumors grow more slowly. And so that's the more proof that these are important mutations. What this gene does is it's part of a complex called the SWI/SNF complex. It affects how chromosomes fold, and it affects how the RNA transcription mechanisms can access the DNA in order to turn DNA into RNA and ultimately protein. And that's called, is controlled by something called the chromatin remodeling complex. There's two types of complexes that you can see here on the slide. The red circle is around what's called BRG1, which is the same name as SMARCA4. And this is a critical component of this complex. And what this complex does, it controls many cellular and biologic functions. You can see them here: migration, signaling, other sorts of hormone programs, and importantly, proliferation. And a lot of this control is regulated through something called EZH2, which will become more important later in the presentation, but this complex is a very important complex for controlling biologic functions. So these are the three papers. One of them is from the McGill group over here on the bottom right. We're over here, and then the TGen Vancouver group. Also, we all published this at the same time last year. So what's new? That's kind of old news. What's new? Well, also there's a hereditary component. So one thing we all noticed is that we can tell where, well, there's a hereditary component to the disease. And before moving on, I'm going to stop at this point and allow Dr. Foulkes to take over and discuss a little bit about the findings of the hereditary component and explain a little bit of the difference between somatic and germline mutations. Will, do you want to take over for a few minutes?
Yeah, sure. Thanks, Doug. So, hello, everybody. Yeah, so we came at this from a slightly different point of view. As a geneticist, I was interested in families with cancer. We noticed there are a couple of these very unusual families shown here in this figure. So what you're seeing here are these four families that we studied. In each case, there was a mother and a daughter affected by the same disease. And obviously, when something's very rare like this, and you see a mother and a daughter together, then obviously you do wonder whether there might be a sort of inherited mutation that caused both tumors. And we found this to be the case, and we found SMARCA4 mutations in all these four families. We went on to, in fact, we've gone on to study 63 patients in total now, where we've been able to test for SMARCA4 mutations, and of those, 27 had germline mutations, so that's 43%. That's actually quite a lot, and I'm not sure whether that's totally representative, but it's quite a big series now. And that, interestingly, 21 of the 27 didn't actually have any family history. So, of course, the question might be, well, what happens if you test the parents? In many cases, we couldn't test the parents because the parents were not available to us. But in 10 cases, we did test the parents, and in 4 cases, the mutation came from the father. In 5 cases, it came from the mother. And in 1 case, it was a new mutation that neither parent carried. And what's interesting is that when we looked at those people who carried the mutation, we only seem to have 3 unaffected female carriers. That is, 3 women who carry the mutation, but don't actually haven't developed the disease. And there are 3 unaffected female carriers who are children or have had preventive surgery to remove the ovaries. So at the current time, we don't know what the risk is if you carry mutation in SMARCA4, but it's likely to be high. We can't really tell you whether it's as high as you'd expect with something like BRCA1, which many of you may have heard of in terms of breast and ovarian cancer. That's probably the most important gene for a different type of ovarian cancer. But it probably is something like as high as BRCA1, although I think one should be cautious about being firm on that point until we've done some more work on that. I think I'll leave it there, Doug, and we'll talk about it maybe a bit later when we come to the end of the slides.
Oh, okay, great. Thank you very much. So some of the new developments, as I showed you, this complex back here has multiple components, and sort of some components are more important than others. And I'll call your attention to this complex and this gene called BRM, which is also called SMARCA2. So what we noticed as we were looking at sort of why these tumors are so aggressive, we noticed that some of the tumors that had a SMARCA4 mutation and lost the SMARCA4 expression, when we stained the tumors for SMARCA2, they also had no expression of SMARCA2. This is very unusual because in other tumors, like the lung cancers that I showed on one of the graphs before, it's been reported that, in fact, when you lose SMARCA4, in fact, SMARCA2 goes up, sort of as what's called a compensatory mechanism to balance the loss of SMARCA4. Other types of tumors will increase the expression of SMARCA2. But in fact, here, and I'm showing you all these immunohistochemistry pictures, we stained 9 tumors here, and they all had loss of SMARCA2. We went on to, we found got a few new tumors from generous people who shared their materials with us. So we also found that SMARCA2 and SMARCA4 were lost. We went over here to some of the tumors that we had frozen material for and also showed loss of both SMARCA4 and SMARCA2. And this is one of the cell lines here, which has a loss of SMARCA2, which is the second band, SMARCA4, I'm sorry, which is the second band, and it also shows loss of SMARCA2 down here. This is a lung cancer line where it has a mutation in SMARCA4, so there's no band over here, but on the bottom part, SMARCA2 is still intact. And so this new finding is that also it appears that SMARCA2 protein is universally lost in this particular tumor type, where there is also a SMARCA4 mutation and SMARCA4 protein loss. And so this is completely unexpected. I don't believe this phenomenon is seen in any other tumors commonly. And so a lot of us are investigating this. One thing, the first obvious question is, well, are there also mutations in SMARCA2? And based on our studies, our original studies, and we went back and we sequenced this gene in all of these tumors, and we did not find any mutations in SMARCA2. We also went back and can look at some of the data from the other investigators who made similar discoveries, and they also did not report any mutations in SMARCA2. So it's not lost because of a mutation. It's lost because some other mechanism that we have not yet figured out, but are working on that, as are other people. Here is actually a paper that also came out recently. Actually, both these papers are really what's called in press. This one you can find online in a Word document form, and the reference is here at the bottom. DOI, whatever that is. Our paper is also what's called in press, and we have the proofs, and so it's not available yet, but in a week or two at Modern Pathology, this paper will be available. This paper is in a journal called Journal of Pathology, and this you can find online currently. This is from the Vancouver, British Columbia TGen group, and so we're all sort of asking the same questions, and they reported a very similar finding that in their tumors also SMARCA2 is lost in all of these cases that have a SMARCA4 mutation. The next question was whether other tumor types can also lose SMARCA2, and so we looked at a number of other tumors that affect the ovary, including what's called sex cord stromal tumors, granulosa cell tumors, ovarian clear cell tumors, some melanomas that can affect the ovary, and in almost all of them, they had both SMARCA2 and SMARCA4 expression that was intact, and so there was no loss of expression in these other tumor types. Finally, here's another paper of ours that's also, I'm pretty sure, actually this may not be quite available yet. It's actually accepted for publication. I'm not 100% sure it's available online, but again, if it's not, it should be out in a few weeks. And here also, we show loss of SMARCA4 in almost all of the small cell tumors up here, and then we look at a whole panoply of other tumor types, and only really one ovarian clear cell tumor and one melanoma tumor that affected the ovary did we also see SMARCA4. And so when it comes to ovarian tumors, the loss of SMARCA2 or SMARCA4 is very specific for small cell carcinoma of the ovary hypercalcemic type. This is an example of some mimics, and so people always ask me, well, you know, there's a small cell carcinoma of the lung, there's small cell carcinoma of the cervix. Is this the same as small cell carcinoma of the ovary? And we never really thought it was the same, but these figures here really show the tumors that are blue and purple, but this is the SMARCA4 expression. And so these tumors that have small cell in their name, they do not have loss of SMARCA4. And so they're completely different tumors, even though the names may suggest otherwise, which is why naming and nomenclature is very important. And I know Dr. Foulkes has some opinions about that he may discuss towards the end of the presentation. Here is actually the clear cell carcinoma of the ovary. The top panel here is the tumor that lost expression of SMARCA4, and this is just another clear cell cancer where it actually was retained, and this is the melanoma. And so while it does lose expression, it's very uncommon, even though we see it in these rare examples. Finally, we were able to obtain one tumor from a very generous patient and family. We were able to actually put that tumor into a mouse, and you can see the tumor here growing inside of a mouse. And this type of resource is so critically important to us being able to test drugs. It's really hard to test drugs on cell lines, and we don't have enough of those either, but we really need the mouse models because that's critical to drug discovery and preclinical work. And so here's an example of the small cell tumor. It looks like a small cell tumor, and it also has lost SMARCA2 and SMARCA4, and it has a mutation here in SMARCA4. And so this is really critical, which gets me to my plea. I mean, the main reason for the webinar is to update everyone on the research, but the secondary aim is to say we really need your help. We cannot advance the research agenda without your help. The community has been so helpful so far, but if anyone's ever having an operation or a biopsy, we really need fresh tumor. We have ways of shipping collection kits anywhere in the world. We have things that keep them at 4 degrees for 96 hours to send them back. So this is our email address, GYN Research Lab, or this address right here. We're happy to email with anybody. We don't really want to answer clinical questions because we're not really allowed to do that, but research questions and anyone can help us with our research, please contact us. We need your help. So moving on to some of the more, some other exciting things. Obviously, we want to make the treatments better. So I'm going to spend a few minutes going over some of the latest concepts in how to treat this disease better than we're doing today. So this is a concept that was proposed by investigators at MD Anderson who are collaborating with our group at Memorial, Sloan Kettering. And for many years, the MD Anderson group has been given a 6-drug regimen that you can see here in AIM-1. We actually don't give this regimen here at Memorial, but my personal opinion is the results from this regimen seem to be better than other regimens that are given. Please keep in mind the data is very limited, and certainly this regimen does not work as well as it should for many people, but it's a good regimen. The second regimen is actually a regimen that's used through what's called the Children's Oncology Group, and it's a regimen that's used to treat malignant rhabdoid tumors, which is a pediatric disease that is related. And again, Dr. Foulkes may make some comments about this at the end. But this is a regimen that's been tested in the pediatric population of malignant rhabdoid tumors and shown to be effective. And so the question is, could this regimen be more effective than the 6-drug regimen? And so we would like to compare the 2 regimens in newly diagnosed patients. And we proposed this to our cooperative group, which is called the NRG Oncology Group. And they accepted this concept over 1 or 2 others that were proposed for us to develop it. So it doesn't mean the trial is approved. It doesn't mean the trial is even open or close to be open, but it means that there's interest in us developing this type of trial. And the principal investigator is Kathleen Schmeller from MD Anderson, who some of you may know, and she's going to be putting this trial together, and I'm going to help her with some of the science that will be done on the trial. We're all very concerned about patient accrual. For this trial, we really need 20 patients in each arm. We know it's a rare disease, and we often hear about people, who have the disease. We don't really know how many patients have the disease who will go on to a trial. My estimate is there's 300 cases a year in the United States, but that could be quite inaccurate, and I don't know for sure. It's very hard to keep track of rare tumors. So one thing this trial will do, if we open this trial specifically for small cell patients, is one very important thing is, can we actually run a trial? Can we accrue patients, and are there enough patients who can fill a trial so we can find out meaningful results? And if we can do that, that's really the best way we can advance the field and make studies better in the future. So this is one thing we're working on. Second thing some of you may have heard about is a study that's recently completed from a company called Epizyme. They did a phase 1 study that I'll explain to you over the next few slides. So as I mentioned, EZH2 also is involved in affecting how the chromosomes open and close. And it's been known that when the SWI/SNF complex, or SMARCA4, is not working properly, it's been known that there can be some dependencies on a complex called PRC2, which can be regulated by EZH2. So the thought is that if SMARCA4 and SMARCA2 or at least SMARCA4 is not working properly, the tumors may become dependent on what's called EZH2. And so if you can inhibit EZH2, that may kill the tumor. And so this company has a drug that targets EZH2. It's highly selective, and you can see in this mouse model here of a malignant rhabdoid tumor that when you give the drug on the purple and blue lines, it kills the tumor. And so with this information and a lot of other information, they launched an early phase trial, and the results were shared about a month or two ago. They enrolled 30 patients, and what's extremely noteworthy is 2 of the patients had small cell carcinoma of the ovary, or now we hear malignant rhabdoid tumor of the ovary. And again, we can discuss the terminology later on. And so, again, these are the responses. This is called a waterfall plot, and all of the bars that go down mean that the tumor is getting smaller. And specifically, these 2 black bars are the 2 patients who had small cell carcinoma of the ovary. So one patient had a very good response. The tumor shrunk by more than 50%. And the other patient, the tumor shrunk just a little bit, but this patient was able to stay on the treatment for at least, I believe it was 25 weeks. And I think the treatment might be ongoing. I'm not 100%. Oh, no, actually, I don't think the treatment's ongoing. And so this maintained the disease in a slightly reduced state for longer than expected. And so that was very promising. This is actually the CT scan. It's a cross-section of a patient's abdomen. You can see a big tumor here at baseline. The tumor got smaller, and then it got smaller since at week 16. And here it actually says the treatment's continuing at week 24. So I may have to restate myself and say that I'm not 100% sure if these patients are still on the trial or not. But certainly this is very exciting. It's a targeted therapy. It has a mechanism that makes a lot of sense. And we see a response here in this one patient, and actually 2 of the 2 patients with small cell carcinoma, they both had a response, and so that's really great and really exciting. So where do we go from here? The company is launching a phase 2 study that's going to be open internationally, and I'm very pleased to report that we have this trial at our center. And the trial is going through our mechanism to open it. I expect the trial to be open in the first quarter of 2016. It's not the eligibility in the trial is not 100% clear to me that we can put small cell patients on the trial. I'm 90% sure that we'll be able to, but we have to look at the details, and we may have to discuss with the company the proper terminology to make sure that small cell patients can go on the trial. I'm told the company is very interested and is fully supportive of having small cell patients go on the trial, and we just have to tweak the study to make sure that there's no problems enrolling small cell patients, but I'm highly confident we'll be able to do that, and I'm pretty confident the trial will be open in the first quarter of next year. These things can take longer than we think. There can always be regulatory agencies and other things that get in the way, and so it wouldn't be a terrible surprise if the trial wasn't open until the middle of next year, but the way it's moving along now, we think it's going to be open by the first quarter of next year, at least at my center. I don't know if other centers have it open, and right now I don't know what other centers will have it open. It may be known, but I don't know that myself. So just to conclude my part, and again, Dr. Foulkes is going to talk about a few things afterwards. This is clearly a fascinating tumor. We definitely need better models. If you know of anyone who's having even a biopsy, there's always an extra piece as long as we can coordinate it and get permission, and that really goes a really long way to help the community. We are developing clinical trials, as I mentioned, that are using these discoveries. I mean, so the discoveries that we've made, my group, Will's group, and David Huntsman and his group in Vancouver, I mean, these really have laid the critical foundation to having new trials that we can now target with drugs that are in existence. And so we're definitely making progress. As always, it's not as fast or as good as any of us would want, but it's certainly a huge change from whatever we had 5 and 10 years ago. So we need your help, again, please. And again, we want to thank everyone. It's a huge community, the Small Cell Foundation, Small Cell Ovarian Cancer Foundation. We're also working with the International Rare Cancer Initiative. It's a, as it says, an international initiative. We have a colleague in Australia who's part of the leadership of what's called IRCI. And whenever we open our trials that I talked about, we can promote them through IRCI to also make sure everyone's aware of them and can get to them. We get funding from a lot of people. This is some of the people, the Department of Defense, the NIH, and our own internal Cycle for Survival. A lot of our funding comes from these places. I think both Dr. Foulkes and I would agree that, you know, we can certainly make more progress the more support we have to do the research, because we know what questions to ask, and it just takes time, effort, and money. And so with that, I'm going to stop. Again, we'll take some questions, and we'll answer. I see a few questions coming through, so we'll answer that. We'll talk about those in a few minutes. But let me open a few slides from Dr. Foulkes to talk about that before we take a break for questions. Will, you want to go ahead? I couldn't put the slides together, so I just opened your slide deck here.
That's fine. Thanks, Doug. So yeah, so this is a bit of a pun in the sense that, as Doug has pointed out, that we do have this disease on the brain. We're thinking about a lot of the time. We're working, we're writing grants, writing papers, trying to understand it, and obviously I endorse everything that Doug said. Next slide, please, Doug. So this is just to summarize some of the things I just mentioned earlier about the germline, i.e. inherited forms of small cell cancer. As you can see, what's interesting is perhaps a bit surprisingly, there seems to be very little difference between the inherited forms of small cell cancer and the acquired forms. In other words, whether you inherit the mutation, either it's a de novo mutation that happened during the process of the development of the child in utero, or whether it was inherited from a parent, it doesn't seem to make much difference on the characteristics of the tumor. The survival is about the same. The stage presentation is, I mean, maybe there's a little bit of difference there, but really I don't think you can say much about it. And certainly the age of onset, again, a bit surprising. The very young ones, as you can see at the bottom, the scale in Figure C, the scale is years up the side, and you can see on that plot that the very young ones below 10 do seem to be hereditary, or germline mutations, but that's about all you can say. The average is not different. Next slide, please. So we were interested in this question about going back to being on the brain, is that we thought that the small cell cancer was actually a bit of a funny name in a way, because they often weren't small cells. They weren't necessarily a carcinoma, which means they come from the epithelium, that's the lining of, like, your skin, for example. And only about 2/3 of cases actually have hypercalcemia. So we wonder whether there's a bit of a misnomer. And we then looked at what the cells did look like. They looked like rhabdoid tumors. In fact, some of the pathologists in years gone by had mentioned that they were rhabdoid. So we thought, well, what other rhabdoid tumors are there? Well, you'll see on this figure here, on the left-hand side in orange, a small cell cancer. We're still using the old term because people are going to take a while to change, I think, and they're going to need more evidence than just me saying so to change their mind. But you can see here's the small cell cancer in orange. Next to it is the so-called ATRT. That's the atypical teratoid rhabdoid tumor, very long name. That's a type of brain tumor that only really happens in children under the age of about 5 years old. Very, very aggressive. Not much treatment, and the treatment that is used is exactly the treatment that Doug mentioned earlier. On the right-hand side, you see a much more common tumor, the high-grade serous cancer of the ovary, in red. And you can see what we're really doing is seeing what kind of mutations do these tumors have, and the height of the bar is essentially the number of mutations. Number of somatic, that's mutations only in the tumor, not inherited mutations. And along the bottom is all the different actual tumors we studied. As you can see, it doesn't take much statistics to realize that the oranges and the blues are much more similar than are any of those two groups to the red. So the a priori idea that because it comes from the ovary, it must be the same, obviously is not true. I'm not sure anybody believed that, but certainly this makes fairly clear that ovarian carcinoma is a very different disease. Next slide, please.
Yep, come in. Okay.
Yeah, so this just makes the same point even more graphically. This is showing when you have whole chromosomes, whole bits of chromosomes being rearranged, you can see it very easily. And again, at the top, you have small cell cancer, then you have the brain tumors, the ATRTs, and at the bottom you have ovarian carcinoma. And you'll see that there's one major line in each case. And the major line for small cell cancer is at where the small cell cancer gene SMARCA4 is. The major line, the blue line on the right-hand side, is where the gene for ATRT, which is called SMARCB1. So it's a very similar gene. And there isn't really a major line for the red one, but if you could see when it's at 17, where BRCA1 is situated. So what's interesting is that again the ovarian carcinomas are much more disordered. So the types of therapies used to treat ovarian carcinoma probably won't work in small cell cancer. Next slide, please. I think the last slide. And just to try to look at this in a bit more detail, we tried to cluster. It's a way of sort of clustering data together. This is looking at methylation patterns of tumors, and that's where, in other words, tumors have marks on them which sort of tells the DNA, as it were, not to be turned on into a protein. It tends to go to sleep, basically. And these marks can tell you quite a lot about tumors. And if you just look at the left-hand figure for a minute, we won't bother with the right one, but the left-hand one is kind of visual in that the red blobs, again, are the high-grade serous cancers of the ovary, and they all cluster together on the left-hand side. The orange blobs are the small cell cancers. So they cluster together as well, but they don't cluster very close to the ovarian carcinomas. They cluster more closely, I think you'd argue, to the blue clusters, which are ATRTs. At the top, you have there's some normal brain, the green ones, and some brain tumors, which the types of brain tumors that are often studied in childhood are obviously very different. They're up at the top. So you could argue, I think, that the light blue and the orange are, again, quite similar. So this, we think, supports the idea that thinking about small cell cancer as a malignant rhabdoid tumor could be a helpful way of determining newer treatments that may offer hope for patients, because if we don't understand the underlying biology, we may go down the wrong road. And I think the very exciting epizyme data that Doug just showed, and this new trial that he also mentioned, it similarly points to a slightly different way of thinking about the disease. So obviously we're very keen to pursue this, and we are doing so. I'm sure Doug and I will be in touch again in the future to collaborate on this. I really think this is a disease where, of course, we all compete, that's part of life, but at the same time we collaborate. And I think, I'm sure Doug and I will be on the phone in the next couple of months and years to talk about our findings and to see how we can work together as well as with the community to advance further knowledge along this line. So just to summarize, we believe that these really are rhabdoid tumors, or that's best described as rhabdoid tumors, because we think that describes the biology better than the previous name. And you could say what's in a name, but a name does, I think, inform a certain thoughts about the way a tumor is going to behave. So I'll leave it there, go back to Doug, and take some questions. Thanks very much.
Okay, thanks a lot, Will. I'm going to just take 1 second here and stop sharing the screens. And I just want to, so if you have any questions, please type them in to the box. Actually, that's just doing a couple technical things here. Just to get back to the epizyme trial, one participant wrote a comment saying that she spoke with the company who said that the SMARCA4 mutation would qualify the person, and I completely agree. The only reason that my comments were a bit tentative is because one version of the protocol that I had seen indicated that SMARCB1 mutations would be qualifying, which is the malignant rhabdoid tumors that Dr. Foulkes was just talking about. And actually, the version I saw didn't actually say that a SMARCA4 mutation would qualify. I hope that the next version, or the version that gets approved, will say that. That was my only slight concern. It says that malignant rhabdoid tumors will qualify, and so as long as we know that that qualifies, that would be sense. So I think it's all going to work out. Just the version I saw just had a couple words that I didn't quite see the mutation. So it should all work out. I'm glad that people think it's also going to work out. We have a few comments that everyone can see, that Dr. Pressy from Cincinnati Hospital is scheduled to open the trial next month and will be accepting small cell patients. That's great. And again, from Cody Weber to everyone, the drug mentioned, the target of NE1, as well as SMARCA4. So any one is SMARCB1, and so that is also part of the SWE/SNF complex and has important functions. I'm not sure I can show you that in a picture, but it's also part of the mechanism by which the complex works. And so there are, each part of the complex sort of has very different molecular functions, but the SMARCA2 and SMARCA4 actually usually are sort of mutually exclusive components in the sense that you need one component to work if the other one doesn't, which is why it's very unusual that both components are lost. I can't really talk any more about the detailed mechanistic differences between SMARCB1 and SMARCA4. Will, do you have any comments on the specific functional differences between the subcomponents of the BAF complex?
No, not really. I mean, it's extremely complex. I mean, we did do a little study which we published recently in Journal of Pathology, where we just sort of tried to picture the differences, and it certainly is interesting that the expression of these molecules, i.e., where they're actually working, because they're obviously present in all cells, where they're actually may be different, and there may be that SMARCB1 has some overlapping functions, but clearly, I think there's only 1 or 2 reports of SMARCB1 causing small cell cancer of the ovary, and so it may be there is some overlap, and they certainly do cause malignant rhabdoid tumors outside the ovary. So why SMARCA4 specifically seems to cause either very, very rarely the brain tumors, but much more commonly the ovary tumors, and nothing much else, really is not at all clear. But no, I think we don't know enough really at all about the differences between these two molecules, but they obviously seem to have very, very similar functions.
Right, and also both functions sort of to perform the activity of the complex, whereas all the other components that I showed are often sort of what I call regulatory components, where it helps to keep the complex together and do other things. Again,
that's right. They have the HEPA domain, and that clearly is sort of part of it. They have the business end of the molecule, if you like. Yeah.
Right, and it is true that the SMARCB1 mutations were originally discovered in the malignant rhabdoid tumors, the brain tumors. They also can affect the kidneys and some other body parts. But there are SMARCA4 mutations that do cause malignant rhabdoid tumors, either the brain or the kidney. I'm not 100% sure. But I don't know of any SMARCB1 tumors in the ovary. I think Will might have mentioned there was one. But certainly it does seem that SMARCB1 is more associated with the malignant rhabdoid tumors.
Yeah, there's one or two. You kind of wonder whether they're sort of somehow not really from the ovary in some way, because it's sort of 98%, A4, SMARCA4, and sort of 1% SMARCB1 and 1% nothing. So I guess we'd have to ask the people who published that to look at that in more detail. I think it's the Huntsman group, but certainly we've never seen a SMARCB1 mutation, and we've done immuno for all of them, and they all seem to have SMARCA4 missing. But whatever the difference between them, obviously it's very important, but we don't know what it is. We have no idea what it is.
Right, and I was also surprised that in your age graph there, like the 9- and 10-year-olds, that really seems to be within the potential age range of the malignant rhabdoid tumors. So I just wonder, are those? I mean, I guess they're involved in the ovary, but are they really malignant rhabdoid tumors versus malignant rhabdoid tumors of the ovary? Because those are pretty young.
Yeah, it gets to be a bit of a, I mean, the youngest case reported was 14 months that I've seen in literature, when it was reported on, I forget where from now, I think Colorado.
In the ovary?
In the ovary, yeah.
14 months, wow, I didn't hear that one, that's very right.
But I mean, obviously, it's uncommon. It's much more common in the teens and 20s and 30s, but there's always a few weirdos that, outliers that make it hard to make general statements, but in general, I think you're right. The very young ones are unusual.
Right, and then Cody Weber had an earlier question that I think he reposted about whether the SMARCA4 mutation is dependent upon SMARCA2 loss. So again, what I mentioned is in other tumor types, when you have a SMARCA4 mutation, the tumor is actually dependent on SMARCA2 compensating. we have tried to look, we've looked at to some extent replacing SMARCA4 in these tumors that have loss of SMARCA4 and SMARCA2, and when we replace SMARCA4, we do not see that SMARCA2 comes back. And so that was one of our early questions. You know, if you restore one, do you restore the other? We have not done the opposite. So I don't know. I don't think they're dependent upon each other, but the tumor might be dependent upon loss of both.
Yeah, that's a very interesting point, isn't it? I mean, certainly when we've tried to put back SMARCA4, the cells just die. I mean, it seems to be, I mean, the tumor cells really don't like having normal SMARCA4, if you like, which makes some sense. The tumor is almost, you could argue, in maybe it's not quite the right term, but the tumor's sort of addicted to having no SMARCA4 around. And once you add back SMARCA4, the tumor cells just sort of keel over. Now, of course, you can't just put back SMARCA4 into a tumor cell. Unfortunately, that's not as easy as it might sound over the phone. If only. I mean, there is something very interesting about that SMARCA2, SMARCA4 balance, and perhaps that's an area where, you know, people could start thinking about what's the balance, what's the real driver of that effect, because it's so exquisitely sensitive, as Doug and other groups have shown, that the loss of the two together is almost a hallmark for the disease. I mean, in a sense, this is the other point to make, you know, for people who are wondering about whether the diagnosis is correct. I mean, for years, nobody knew how to make this diagnosis. You'd have to send it to Scully or a few experts like Glenn McGill, or a few people around the world who'd seen who trained with Scully, basically. It was Scully and people who we trained who knew what this thing was. And now, essentially, if you get these SMARCA4 and SMARCA2 antibodies working, which is not necessarily trivial, but if you get that done and you stain a very unusual ovarian tumor and it shows no staining for these two proteins, that's a diagnosis. So I think that's a real change for pathologists, that they don't have to spend days staining it for everything under the sun, trying to rule out desmoid, desmoplastic, whatever tumors of the whatever, and all that kind of crazy things it can mimic, but it tells you straight away. So that's a huge change. But I do think, as Doug said, that there's definitely a sort of a lever there to get into the treatment aspect of things.
So one other comment that came through from someone was a question that 100% of the individuals, of the tumors in the original study, tested positive for SMARCA2 loss, and so that's correct. So nearly all the tumors, you know, in our study it was all the tumors, but of the collected literature, nearly all the tumors have SMARCA4 mutations. And now my group and David Huntsman and his group, we've both shown that all of the tumors, with maybe one exception, also with one exception, also have SMARCA2 loss. And in fact, we had one tumor in our original report that had a SMARCA4 mutation, but the protein was not lost in one tumor, and that's actually the same tumor which doesn't lose SMARCA2. And so that's really fascinating. It's also so interdependent. So yes, almost all of the tumors also lose SMARCA2, which is the question someone was asking for clarification.
Yeah, interesting. Yeah.
And then one other question we can see here is, where does EZH2 fall in the biology? So again, that's to the PRC2 complex. And so the idea is that if the SWE/SNF complex is not working properly, then EZH2 will become the PRC2 complex will be important to take over the function that was maintained by the SWE/SNF complex, and PRC2 complex is under the control of EZH2. And so the idea is that if you inhibit EZH2, you actually are inducing what's called some form of synthetic lethality, where you now can suppress the PRC2 complex. We actually tested an EZH2 drug, a different EZH2 drug, and actually didn't see that it worked. We haven't completely finished that testing, and we haven't tested the Epizyme drug ourselves in our own models, but we're doing that to make sure we can also see these findings, and we can do all sorts of interesting studies when we look at that.
Yeah, and of course the question of whether the effect will be sustained, and whether you showed there that some of the tumors actually do acquire mutations later on in your patient-derived model where you grew the tumor in a mouse. Clearly there were some mutations. I think I saw on the screen there were some mutations that perhaps you wouldn't have expected initially. So there may be some progression to a different type of tumor that becomes resistant to the Epizyme drug. But certainly it's it's the first hint, isn't it, that I think that there could be something in this therapy that previously we didn't know about.
Yeah, you're exactly correct, and we have not been able to, like I said, we just haven't got around to it yet. Well, our first attempts at using EZH2 inhibitors were not successful. We really haven't, looked into that in enough detail, just mostly because of time and also resources, meaning model systems. And so it's a good question as to whether inhibiting that would increase SMARCA4, SMARCA2. We have not looked at that. I don't think the other groups have looked at that as far as I'm aware. People are looking at the role of EZH2 in reducing tumor size, but I don't know if that restores SMARCA2 or SMARCA4.
Right. No, we're doing similar sorts of studies on different kinds of, in collaboration with colleagues at McGill, with different types of inhibitors, and obviously everyone's looking at this in different ways, and that's the way things progress. You all have a different sort of angle on something, and that's how you get forward. But certainly it is a new angle that we're all interested in exploring.
So if there's any other questions, please feel free to type them in. Otherwise, we'll wind up soon. One thing we didn't talk about is immunotherapy. Oh, there we go, immunotherapy. So, yes, exactly, nivolumab. So I was just going to mention that there was there's one patient who may have had a response to nivolumab. We're just beginning to look at this, and so I won't make any comments right now as to what we're finding. I'm not an expert in immunotherapy, but I do have some people here that I'm working with, like Will's worked with his people in McGill. So we're beginning to look at this. It seems like it could be promising, and it would be so wonderful to have multiple options here. And so I don't have any, specific comments that I'm prepared to share regarding immunotherapy in this particular tumor type today.
No. But it's the kind of drug that's basically going to be tried for almost everybody, because the results have been impressive, haven't they, in some unusual situations like lung cancer, when you weren't perhaps expecting to see such prolonged responses. And obviously a disease like this, where the outcome is generally not always great at all, many people will consider using something like this, even though the evidence for it, the scientific rationale for using it isn't completely apparent, but I'm sure, you get surprises, and I'm sure it'll be something people will consider.
Okay. Well, I think this has been excellent. I want to thank everyone for being on the call. I'm just going to take 1 second now to, if I can do this. Yeah, I'm going to take 1 second now to stop the recording.